Good morning, everyone. Do you hear me well? Nice. So I am pleased to share my thesis work today about beneficial effect of the environmental enrichment on transgenic mouse model of FTD-ALS, supervised by Dr. Anaëlle Boutillier at the LNCA in Strasbourg. So first, I would like to talk about the ALS-FTD continuum. So, as you may know, amyotrophic lateral sclerosis and frontotemporal dementia are two distinct pathology, but one is more related to motor symptoms and the other one to cognitive symptoms. But over the past decades, so researchers discovered that they shared disease spectrum. We've shared clinical imaging and genetic features. Like you can see here, we have a lot of genes in common between ALS and FTD, like TDP43 or FUS. And now how we modelize this in the lab. We have a heterozygous knock-in FUS delta NLS mice model, where the NLS is truncated. So normally FUS is expressed into the nucleus, but when delta NLS is truncated, we have a mislocalization into the cytoplasm. And this progressive model show gliosis at 1 month, cognitive deficits that were highlighted in our lab by spatial and memory testers, as well as epigenomic and transcriptomic alteration. At 10 months, they show motor impairments, and at 13 months, anomaly of the neuromuscular junction. I will take advantages of this progressive model of ALS and FTD to study the cognitive impairments as well as the modulation by environment. So this is another side of my project. So enrich environment into people, it's modelized by 3 components: the physical activity, social interaction, and novelty, that have beneficial effect on the brain, the metabolism, the cardiometabolism, and the neuronal function. And in rodents, in many animal models on neurodegenerative disease, we have EE that enhance hippocampal dependent condition by triggering synaptic plasticity genes and LTP, the production of the BDNF, memory formation, as well as neurogenesis. Oh, sorry. How do we modelize this in our lab? We have 2 different accommodation conditions. The standard environment, that is a classical cage where the mouse are housed per 2, with no other stimulation than a nest and food and water ad libidem, versus the enriched environment modelized by Marlowe cages. Those are big cages with 2 different components and 2 different floors. So the food and the water are separated but by one-way door, and the mice have to climb the ladder, cross the maze that is changed 2 to 3 times a week, and then slide down the tunnel to have access to the food. This enables us to have the same treatment for all the mice. And so social interaction is modelized by the number of mice, the physical activity by the running wheels, and the novelty by the maze. The objective of this study is to test the enrichment effect on cognitive functions by behavioral approaches like actimetry, spatial memory, and object memory, but also to understand the underlying molecular mechanism by RNA sequencing and cut-and-tag sequencing. How do we do that? We have 2 different cohorts, one for behavioral analysis and one for biomolecular analysis. After 5 months into Marlowe cages, EE, or standard cages, SE, we put the mice into behavioral analysis, like actometry, Morris photomaze, and object in place. And for the cohort 2, it's exactly the same, but they are euthanized to do RNA and cut-and-tag sequencing. So first, I will a little bit talk about the behavioral results. We did some object in place, so it requires the mice to associate an object with its place. And for the protocol, we have 2 different phases: acquisition for 15 minutes with 2 different objects, 5-minute rest into the house cage, and then probe test 15 minutes with 2 of the same object. I scored the time exploring the object to calculate the index of retention. Here you can see that the fused mice are really deficient in this test. But we nicely recover memory function with enriched environment. So what about the transcriptomic now? I did some RNA sequencing. After DSEQ2 treatment, we discover a common effect of enrichment. So we can see here that we have more upregulated genes into the 2 genotypes in EE condition. Then we crossed these 2 upregulated induced genes, and we obtained 26 genes in common, and among a dozen are related to immediate early genes. And we can interestingly see that we have a very big induction of immediate early genes in both genotypes, wild type and fused mice. But what about the disease-specific effect? Is there any? Yes. And as you can see here on this graph, we have a nice induction of neuronal and synaptic plasticity pathways only in the fused mice in response to EE. We have some downregulated specific genes related to extracellular matrix genes that I don't show here today. Before going into the epigenomic results, I will a little bit explain what cleavage entre targets and tegmentation, shortened cut and tag, is. First, I will talk a little bit about nucleosomes that are composed of histones, and on those histones you can see that we have epigenetic marks. Those epigenetic marks can be active or repressive. As you can see here, those are the marks that I tested during my thesis. So for the protocol, we have an antibody that targets an epigenomic marks that is coupled to a transposase that can cut the DNA fragment that is tagged. Then after we amplified those DNA fragments, sequenced it, and aligned it on the genome. This allows us to identify genomic loci that are enriched or depleted in specific histone marks. For a matter of time today, I will only present H3K27 acetylation. So here are the results for the cut and tag. We can nicely see that we have an increased acetylation in both genotypes, fused and wild-type genes. And those genomic loci are related to synaptic learning and memory function, LTP, as well as cytoskeleton in both of the genotypes, and especially in wild-type into mitochondria. But what about the common effect of EE? We can nicely see here that we have an increased H3K27 acetylation on genes related to synapse organization, synapse morphogenesis, cell morphogenesis, and chemical transmission. That is significantly in wild-type mice. But how about the fused mice? We can see, that we have a partially recover of the acetylation. Sorry, I lost the pointer. On certain genes here, like that. And we identify into fused EE and fused SE data sets some mRNA metabolic processing, RNA processing specific, this is specific effect of EE after the GSR analysis. And this is very interesting because fuse is a mRNA binding protein. So for the beneficial effect of EE at the behavioral level, EE restored memory in fused mice. At the transcriptomic level, we have an increased neuronal activity associated gene transcription, selectively impacted neuronal and synaptic plasticity genes transcription into the hippocampus. And at the epigenomic level, we have an H3K27 acetylation enrichment, an increased chromatin opening on many genomic loci associated to neuronal plasticity genes in both genotype, fused and wild type, and further on, this is specific genomic loci on mRNA processing. As a non-pharmacological therapeutic option, EE improves plasticity and cognitive function in FTD condition through epigenomic changes. I would like to thank you all for your attention, and I would like to warmly thank the ALS Foundation for their generous support in funding the 4th years of my PhD.
Thank you, Anaëlle. It's time for questions.
So it's a great result because that could reinforce the idea that social interaction may be effective for patients with FTLD, and so because we have difficulties to demonstrate that.
Yes.
In humans, and so it's interesting finding that.
Yes, this is very interesting because we know that enriched environment is very beneficial for everyone, like a patient or not, but we don't know how it worked. So here we can see that through the change of the environment, we can change all the genome, like the epigenome and the gene expression, and so to enhance memory function, but also neurogenesis and mitochondrial function too. And yeah, maybe it will enable us to identify some targets that could be like after modelized into treatment.
And if you modify your the environmental stimulation and you come back to the previous way, do you observe the modification, or
what
to have
we
a crossed, for instance, methodology on the?
So what we see here is that we have the two cohorts. One that have like here time with behavior, and after we can see that during this time, that is almost 2 weeks, we preserved the EE effect. So here we can still see that it improves memory function. Here we cannot see. It would be interesting also to have longer time where they are not enriched anymore, and to see if the epigenetic and transcriptomic changes are still there.
I have a question. So if you compare the EE and EE, actually, in control versus FOS, yes. In this case, in the FOS mice, do we agree that you would have maybe some kind of normal loss because you take them at an age where they should already have, or they are before any loss or modification, even if you don't have yet loss, the FOS by itself induces some neuronal modification. Also, all the changes that you see, do you know if the cell type is the same and in the control versus the FOS, and how can you make the difference between what is linked to the FOS directly from the disease compared to, because in control you don't have any loss of neuron or anything happening there compared to the FOS?
So normally we use the mice at an early stage of the pathology, and as it's a progressive mouse model, we don't have really loss of neurons. Normally it's preserved, but we have mild neuronal impairments that we can see like into the, here with the behavior, that we have a loss of cognitive function into the FOS that is caused by EE. So normally we know that the effect also is mild into the fuse because of the pathology, and better here in the wild type. But
But do you see that you rescue basically what, because here you rescue the phenotype, but if you compare in the wild type and the FOS in like normal condition, you would see that you have modifications because the neurons or other cells are modified by the pathology of FOS basically. And here you rescue it, but do you also rescue what is usually coming from FOS in your RNA-seq actually?
So what we can see here for the disease-specific effect is that we have a rescue on the mRNA metabolic processes. So, and we know that fuse is super important for all the mRNA processes. So here we can see that we rescue it, but then after into the RNA-seq data, we can see for some that we have like induction of immediately genes that triggers synaptic plasticity. So it triggers in wild type, but also into the fuse. And here we can see a nice induction. So I think, yes, we can, like on a certain amount of genes, reverse the transcriptional activation.
Thank you.
Thank you very much for your presentation. I have maybe a naive question, but in case, maybe you know, in case of presymptomatic cases in human, is there a recommendation of EE? I mean, stimulation, like you described in mice, but for human, what would be the recommendation?
For me, everybody should be enriched. But for the patient, it can be transcribed by like talking to them, doing some social game, for example, or it is shown in ALS if you do a mild physical activity, it can enable longer and delay the motor symptoms.
Sorry.
So yes, so maybe it's not that it should be more implemented into the things that we say to the patient, like when they go to the doctor, they should say that you have to do a little bit more of social interaction or physical activity to maintain you, and that is beneficial after for your neuronal functions.
So very important study shows that animals and experiments are very prone to artifacts if you do not do them properly. So one question is, when did you separate the mice? I didn't get, maybe I didn't pay
So
much.
after 1 month, after weaning.
So they were very young.
Yeah,
so
they were very young.
So it might also change or influence neurodevelopment or like maturing of the brain even, if you...
Yeah, true.
So I wonder, so if it's not only changes epigenetic, but also the wiring of the whole brain, and I would be interested, do you see behavioral deficits with this mouse line, like FTD phenotype?
Like, so for the first part, we see some neurodevelopmental changes, like linked terms. Like here, you can see that we have nervous system development, so it doesn't shock me. It will be the case. And do we see some FTD phenotypes? Like we have some cognitive impairments, but we didn't test it, like the social interaction. So I don't really know for this mice model.
This would be interesting if you can prepone or postpone it with your with the better cage actually. No, yeah.